On behalf of New Zealand Endodontic Society, I would like to welcome all the participants to our September webinar. Just a few housekeeping rules. Please have your audio muted at all times, such that there's no disturbances during the presentation. Also, if you could submit any questions in the chat box as you go along, and if you could just make sure that all the questions that you are asking are pertaining to these, the webinar topic. They will be answered at the end of the webinar. I will also announce the CPD number at the conclusion. So now I would like to introduce our speaker tonight, Professor Robert Love, who has kindly accepted to speak. Professor Love is Dean and Head of School of Dentistry and Oral Health at Griffith University, a specialist endodontist and chair of the Australasian Council of Dental Schools. He teaches undergraduates and postgraduate endodontics, and his prime area of research is the mechanisms involved in dentine colonization and infection, with current research focused on relating endodontic infection to immunological response, development of antimicrobial peptides to manage endodontic biofilm and infection, and development of keratin-based biomaterials to simulate regeneration of pulp and periradicular tissues. Professor Love has published and presented extensively and was awarded the IADR ANZ Division Oral Biology Award and Alan Docking Science Award. He has been involved with numerous professional organizations, including the NZDA, New Zealand Endodontic Society, Australasian Dental Association, Asia Pacific Endodontic Confederation, IADR, ANZ Division, and served on editorial boards of a number of journals, including the International Endodontic Journal, Dental Traumatology, and Scientific Board Journal of Endodontics. He has a long history of service to dental regulatory organizations, including chair of the Dental Council of New Zealand and roles with the Australian Dental Council, Dental Board of Australia, the National Dental Examination Board of Canada, and the Dental Council of Hong Kong. He was appointed as an officer of the New Zealand Order of Merit for services to dental accreditation, education, regulation, and research. It's a pretty impressive bio you have, Robert. And once again, thank you very much. I'll let you now speak.
Thanks very much, Dikesh. I'll just take that down there. And thanks for asking me to talk. It's really good to be able to talk to the Society again. It's been a little while since I was involved with it. First slide here is if you recognize it. This is sort of in the Mackenzie country, looking out pure east, running down that valley. And when you hit the coast, there you do a sharp turn to the right and you'd get to Oamaru. So now that's not moving again, Dikesh.
Should be. It's in your control, Robert.
Yeah.
Try the arrows.
Yeah. Pushing all the arrows. Let's try that one.
Yep.
Okay. Good. Topic I was going to talk about was endodontic biofilm, a bit of microbiology, which is not the most exciting part of endo or dentistry, but I'll try and make it the clinically significant, so that we will be able to see what the applied aspects of biofilms are to management. This is my slide that I tend to put up all the time. The middle part's my making, but the images on this left-hand side and the right-hand side, these are taken from Miller's 1890 textbook, who was looking down the light microscope at various slices of teeth, enamel dentine, pulp tissue, etc., and was drawing what he could see. And you can see on the left there that that's the internal tubules, and you can see that they've got some material in it, which would be bacteria. And that's not dissimilar to what you would see on a scanning electron microscope slide or a modern day light microscopy slide. And on the right hand side, he was drawing the bacteria that he could see. And again, there we could recognize what he was drawing in various things. He was drawing bacilli, and he was drawing cocci. So it wasn't nothing's quite new in the microbiology field when it comes to the basics, but there has been some advances that are of clinical significance. So pulp and periapical disease, it's a biofilm disease, and then you've got an inflammatory host reaction. Okay, so that's not unusual for us in dentistry. You would be able to say exactly the same for periodontal disease, the exact same two concepts that were happening. So what we'll do is we'll just have a little bit of a look through both those aspects, and then we'll look at management of it. So a biofilm is, I've got it down at the bottom of the slide here. I like to think of biofilm, it's a mass of cells that are functioning like a tissue in the human body, for example. In a muscle, you've got all your muscle cells, they're all working to do a common good. Liver, you've got all lots of different cells in your liver, etc., and they're all working together for a common purpose. So the biofilm is a mass of cells also functioning like that. They're functioning for a purpose. Biofilms can only adhere and start to form in wet environments. And of course, in dentistry, oral cavity, in the pulp, periodicular tissues, it's always wet. So that gives them an ability for bacteria to start to stick to surfaces. They tend to also have a bit of a glue-like substance to anchor them. You can get single bacterial species, and for example, one of the ones in endodontics, which we won't consider, but is Enterococcus faecalis. It can live as a single biofilm species, but most often it's a multi-cell biofilm with lots of different types of bacteria, as well as maybe fungi and things like that incorporated into it as well. So these biofilms, when they're functioning like a tissue, they're carrying out a variety of reactions. And as you know, the biofilms that we all have, a lot of them are benefits to us, and some are detrimental. So these are our major ones in dentistry, isn't it? The dental plaque. So we've always called, from a long, long time, we've called dental deposit a plaque. But really what we should be really calling it is a dental biofilm. And of course, dental caries, and then it runs on to infected root canal systems, infected periodicular tissues, and also periodontal disease. So there are two classics when it comes to biofilms. So we'll look at a little bit about the influences on what forms a biofilm, what makes it form. If you're really, really keen on getting into endodontic microbiology, there's the textbook, Endodontic Microbiology, would be one to look at. But if you just wanted to hone in on the endo, bit of micro, bit of karyology, microbiology, and biofilm structure and all that, you could go to chapter 3 there. The second edition of that textbook. I wouldn't bother with the first edition. I authored that myself, but on the second edition, I was lucky to get Ann Tanner from up in the US to collaborate, and that chapter is really good from having Ann's contribution there. So a biofilm, there's a couple of things we need to consider when thinking about a biofilm. One thing is how cells are in an environment. So as in this image that I've put up here, I've labeled some round blue cells as planktonic cells. So planktonic cells are cells that are floating around in the liquid. So it could be saliva, gingival crevicular fluid, intestinal tubule fluid, etc. They're floating around. The cells that then start to form the biofilm are different. So there's a difference between the activity of those cells. Essentially, a planktonic cell is easy to manage and easy to kill, so to speak. While they're in cells are in a biofilm, we'll see later that they've got specific defense mechanisms that help them stay alive. So, you know, if you've got the reps coming around and say there's this great new endodontic irrigant that kills 99.99% of bacteria and fungi and viruses, etc., you just have to look at the methodology and see if is this on a biofilm or is it on planktonic cells. If it's planktonic cells, it doesn't carry as much weight as a biofilm. So the initial aspects of forming the biofilm, it happens the initial parts happens very quickly, seconds to minutes, where those planktonic cells start to adhere down to a surface. Okay. Now, and this diagram, that's called a substrate. So that could be enamel, dentine, epithelial cells, anything like that, amalgam, composite. That could be substrate. But over that substrate, there's always a conditioning film. And in dentistry, we've always known for a long, long time about the acquired pellicle, which is formed very, very, very quickly after a tooth surface where it's cleaned and saliva interacts with it, and then all those nice big glycoproteins, etc., stick onto the surface by electrical forces. So that acquired pellicle is a conditioning film. So the cells actually stick to that conditioning film in the first instance, rather than to tissue or enamel or dentine. And initially, thereby, again, electrical type bonding systems. So they're not very strong, they're relatively weak, but they stick down initially. And then after a bit of time, specific bonding chemicals on the in the cell surface can interact with the big proteins, etc., in the conditioning film, and then start to form a chemical, more of a chemical bond down, sticking onto the surface. So that happens within seconds or minutes of a surface being exposed to planktonic cells. I've drawn these cells as round blue cells, and that's to represent gram-positive cells, gram-positive cocci. So essentially, in our field, it's streptococci. And they're the initial cells that start to bind onto surfaces, start to form a biofilm. So they're called the primary colonizers. And again, they've got the chemical structure in the cell walls, etc., to start forming those initial bonds. Within ours, more cells start to aggregate together onto that developing biofilm. Ours, this is not a biofilm yet. This is just a coaggregation of cells. And at some stage it will get more complex. But at this stage, after a few hours, I'll demonstrate here, again, it's all just primary colonizer cells coming in there, sticking to the conditioning film, but also adhering, coaggregating with each other as well. And when you look at a biofilm, even though it's bound onto a surface, most of the cells are not bound to the surface. Most of the cells are actually binding to each other. And it's only a few cells that actually do the binding to a surface. So that starts to build up a collection of cells. They're not doing anything at this stage, but they're just trying to maintain their growth, etc. So to fully form a biofilm, there's a number of things that have to happen. One of the major things is nutrition. So those cells that are starting to form the biofilm have to get nutrition. If they don't, then the biofilm it'll fail. It won't carry on. So in oral cavity and dentinal tubules and dentine and pulp and necrotic pulp, etc. There's lots of nutrition available to cells to get to start keep growing. The Tustig and Van der Hoeven paper, it's a perio paper, but it sort of lays out quite nicely the types of cells that can be present in a biofilm during its development. So again, the initial cells, those primary colonizers, they're saccharolytic bacteria. So they eat carbohydrates. Okay, so carbohydrates are the most readily available form for nutrition. So they're the first ones. That's one of the reasons why they're the first ones that can be able to attach and start forming a biofilm. They start to use carbohydrates. Then when the carbohydrates start to run out, other various proteins are hydrolyzed to start to give amino acids into the system. So this is actually done by the cells. Bacterial cells can actually start to break down the sort of proteins into amino acids, but also the host defense mechanisms will cause that as well. The big proteins, etc., will start to break down, particularly if we're looking at a periradicular infection. And then you get sort of proteins that hydrolyze. That means other bacteria can then start to get involved in the biofilm, particularly more fastidious bacteria like P. intermedia, F. nucleatum, etc. And then the final phase is when there's no carbohydrates left, but there's just lots of broken down protein and lots of amino acids, and then that starts to favor the really fastidious cells like P. micros and so the porphyromonids and cells like that. So that gives a bit of a course on how nutrition takes effect on the biofilm formation. Other things as well that the nutrition is that, again, various cells can actually supply important nutrients to other cells, so that's helpful. But the other major thing is that there are bacterial associations. Okay? So there's strong positive and there's also negative associations between bacterial species. So Sundquist actually started to describe this way back in the 90s. We weren't talking biofilms back then. We weren't talking how they were formed, but he was identifying what he could see, and he saw that whenever I saw say for example P. micros, always saw C. rectus, always, and vice versa. When I would see another cell, wouldn't see another particular type of cell. So there's positive and negative interactions going on between cells as the biofilm starts to form. Redox conditions are important, and again, the biofilm will start from a facultative anaerobe strep gram-positive type scenario, and then it will transition through where there is more of an anaerobic environment around, and then again that allows the fastidious anaerobes to be more dominant, like porphyromonids, etc. So the third stage of the development of the biofilm is when all these cells have been able to come in and start to form a big coagulation of various cells. Okay. Now that takes days or weeks to happen. It doesn't happen within hours. It takes days or weeks for that to get stage. So the diagram over there is showing when the biofilm formed. It'll have an extracellular matrix, so some sort of glue, but that also forms an air capsule to protect cells. And it'll have different types of cells involved in it. So we've got red, purpley red cells, so gram-negative cocci, etc. The green lozenge type cell, that structure there is trying to depict Fusobacterium nucleatum. And that is a cell that is pretty much found all the time in our biofilms. And it's called a bridging bacteria. Marsh coined the term a while ago as a bridging bacteria. What it can do is it can bind two types of cells or more that would normally not bind together. So for example, on the left-hand side of that diagram, we've got a strip, which is bound into the conditioning film. Fusobacterium nucleatum is bound to that, and then there's two yellow cells there, so those yellow and blue cells would normally not bind or interact with each other, and so the yellow wouldn't get into the biofilm, but because F nucleatum is there, they both can stick to that, and they can get introduced into the biofilm. So this is a big consortium of cells, again, starting to work and as a tissue, as I said. So they do various things in the biofilm. So one of the things that's always mentioned is about quorum sensing. Essentially, quorum sensing is that mass of cells in that biofilm can actually tell what's going on in its environment, and it can start to limit its growth. It can tell it hasn't got enough nutrition, for example, so it will start to limit its growth, etc. So it's an important aspect of biofilms. It's a way of the cells talking to each other. So there's a lot of metabolic communication going on between cells and lots of genetic interchange as well. So this is all working away again to form some sort of function for that biofilm. But the important thing there is it takes days or weeks to form a biofilm. So for example, if we had a patient who unfortunately had a trauma and upper central incisor fractured, MID fracture, pulp exposed, and couldn't get to see you for 1 or 2 days or even longer, there won't be a biofilm formed on that exposed pulp. There could be a structure like B formed, where the cells are sticking and starting to start to form a biofilm. But after that short period of time, there won't be a biofilm. So there has not been a lot of infectivity action, shall we say, going on in that exposed pulp, and that's why it's quite easy, to manage that, and you don't expect to have any problems from that point of view. If it was 2 or 3 weeks later, then you've got a biofilm on it, and you'll have quite a significant inflammatory response going on in the pulp. But it doesn't. So the important thing is that biofilms take a long time to get up and going. So they're the general properties of a microbial biofilm. There's lots of the various things there, and as I mentioned before, Sundqvist, done an awful lot of work in endodontics. His microbiology papers, he was describing these things in an endodontic scenario, but it wasn't all put together that we were really dealing with a biofilm. So I mentioned before, what's the advantage of cells living in a biofilm? It's basically that they can be protected from the external surface, from the external environment, from host defense mechanisms, trying to eradicate the bacteria, or from medicaments that we might be using to try and manage a biofilm, for example, a topical antimicrobial, or even systemic antibiotics. And so the mass of the biofilm, essentially, it can deplete a medicament in its peripheral surface. So the outer surfaces sort of take the hit, so to speak, and then it can leave the deeper biofilm, the deeper cells in the biofilm still vital, and they can then regrow again. Okay? The other aspect about that is that that insult to the peripheral cells, etc., it can also make those residual cells become more resistant, become tougher to kill. They can start to upregulate other mechanisms to make themselves, to be able to defend themselves a bit better. And that's one of the main causes of antibiotic resistance from systemic antibiotics. They're used for a biofilm infection, and maybe not used long enough because patients say, I'm feeling better now, I don't need to worry, I don't need to do my full length of script. And the biofilm's still there, and those residual cells can form a resistance to that antimicrobial. So they're there to big advantage to the cells. What we consider with a biofilm is about its pathogenicity. These are just a couple of slides that show a bit of invaded dentine, and slide B is a transmission electron microscope of a biofilm in the dentinal tubule. As I mentioned before, there's a few cells that stick to the surface and all the rest are sticking to each other. But what we consider now with biofilms and the management and how to consider the management is to think that that community, that biofilm, how does it become pathogenic? So we'd assume that most biofilms, as they're growing up, they're not pathogenic. And as I mentioned before, there's we've got lots of biofilms that we have to keep healthy. So a lot of the research is what switches a biofilm from being healthy to pathogenic. And there's a lot of work in the medical field to looking in the areas. In dentistry and dental caries, we worked this out a long time ago. What makes a biofilm turn pathogenic? A dental plaque, a supragingival plaque biofilm. How do we make that become pathogenic? We feed it sugar. We don't disrupt it, and we feed it sugar, and those cells will start to go cariogenic, and therefore then they'll start to form dental caries. So we've known that for a long time, and it's the basis of we tell patients to brush your teeth, floss your teeth, etc. Disrupt that biofilm, stop it growing, and don't feed it sugar. In the periodontal area, I don't think it's still not quite as clear why a biofilm might be a bit more pathogenic, but I think it's about, most in that case, again, it's about time. It's that subgingival crevice. There's the nutritional aspect going into that area from broken down proteins and then getting into your porphyromonas and various things like that. But it's not quite as clear cut why a perio biofilm, so to speak, goes pathogenic. When it comes to actual bacteria and endodontics, I'm still sticking to Sundqvist. We can look at all the papers about PCR techniques and your genetic techniques to identify various species in infections. But when it boils down to the practical nuts and bolts of it all, the ones that you can grow up in the laboratory are the ones we probably should be worried about. And I just break it down into those sort of general things there that Sundqvist said are the major cells involved with an endodontic infection. So here's a couple little diagrams of a biofilm. So a little tuft of biofilm at the top left there, etc. But the image on the right, so this is at the bottom portion of the image is the coronal aspect, and top portion is the apical aspect, and that's the root canal. And we can see in that lower half there, we can see a darkly staining mass. So that's biofilm. It's got spaces in it because anytime you do tissue processing, it all breaks up, and you never get. It's very hard to get a nice homogeneous look about things. But that's a biofilm working its way down the root canal there. So it's the pulp has been fighting against it, but it's losing a battle. At the top portion there, that tissue in the pulp chain and the root canal system there is so that is pulp tissue. If you look really hard, which is difficult, impossible in this image actually, but you'll see that it's pretty much non-vital. It hasn't got very good structure with its collagen. Fibroblasts are not really there. There'll be blood vessels are still in there, but the rest of the tissue is starting to become non-vital. The bits in between there are the inflammatory cells. So if you could see them clearly, you'd see that they're masses of inflammatory cells which are coming out of that pulp. Yeah, so part of the inflammatory process, those cells are coming out of all those blood vessels, and they are interacting with that biofilm to try and manage that infection as it's working its way down the root canal. This is just taken right at the apex of a tooth. And get orientation there, the purple stuff, that's a stained biofilm. And then you'll see that on there's interaction between tissue, looking tissue up here, and then this white area here, and that white area is cementum, and this is the dentine. So that there is this cemento-dentinal junction, this point here. You can see very nicely delineation between the dead pulp tissue, infected dead pulp tissue biofilm, which is the darkly staining blue stuff. It's well delineated here, well delineated between this tissue here. Okay, so this is pulp. Sorry, this is periodontal ligament, and this is a mass of bit of inflammatory cells and bit of periodontal ligament as well. So this shows quite nicely where the endodontic biofilm technically ends. It's right at the cemento-dentinal junction, because any tissue that's attached to cementum is periodontal ligament. It's not pulp. And the periodontal ligament is really good at looking after itself. It's if you look at the square millimeterage of a periodontal ligament, it's massive. So it's a very vascular and it can look after itself. So that's what's happening down at that area. So that's a good microbiological indication or marker of where we're finishing our root canal preparation. We're finishing at that cemento-dentinal junction at the apical constriction. So that brings us on to a little bit of immunology, which won't be too much, and the bacterial immunological links with what's actually happening when these biofilms form. So the major things is that the host cells, our cells, and other animals, there's a bunch of cells that can recognize things that are called pathogen-associated molecular pattern. So essentially, it recognizes chemical sequences, yes, molecular sequences. Yeah? So that could be a structure on a cell wall of a bacterial cell wall, or it could be some sort of other things being released from a cell. Yeah. But those cells are able to recognize those molecular patterns from the bacteria. So same with yeast, but I'll just say bacteria. And the host mechanisms that are detecting those molecular patterns are called pattern recognition receptors. And the most important one for our area is one that's called toll-like receptor. There, and that little diagram below there just to show that it's pretty much structure. It's got a bit of the structure that sticks out the cell wall, a bit that goes through the membrane, and then a portion that is within the host cell. And they're actually been shown that the toll-like receptors are really important in both the innate and the adaptive immune system. So they actually function over all that area there. So in the innate immune response, that those cells, in particular cells, we've got here dendritic mast cells or macrophage, actually recognizing a new pathogen. In this case, a bit of a bacillus or a bacteria at the top there. So it's recognized that as new, and then it's able to fire off the innate immune system to start the immunological response going. So cytokines, etc., are all going to be starting to be developed to start the inflammatory process. But it also works again in the adaptive immune response. So it's again, it's already pre-primed because it's already seen that pathogen before, or that PAP, that sequence, and therefore it can fire off the immune system right away, start to get into the T cell population response to it. So they're very important structures on cells. We've got 11 down here. I think there's about 14 of them that have been identified. We're first identifying the Drosophila fly, but they've been shown to be in all sort of animal species there. The important one for us in endodontics is toll-like receptor 2 and toll-like receptor 4. So essentially, toll-like receptor 2 recognizes gram-positive bacterial sequences. And toll-like receptor 4 recognizes gram-negative sequences. So in that 4 there we've got LPS, so lipopolysaccharide, which is in the gram-negative cell walls, particularly released when the cells die. And we've known a long time in endodontics that LPS is associated with inflammatory responses in pulp and periapical tissues. So they recognize that, so the toll-like receptor recognizes that, and then a big chemical reaction works through them. And then within the cell wall, it starts up a big cascading chemical reaction, which starts the cells to form all the inflammatory cytokines, etc., that are important to start giving messages to the cells to start the immune system going. So, are they expressed in pulp and periapical tissue? It's expressed in all soft tissue. So we did a little bit of work with Charlene and Eric, who are both endos in Sydney now, and we had a look at periapical tissues in this case, and just to see whether the cells within a periapical tissue were expressing toll-like receptors, just to show a bit of a definitive link that bacteria are involved with infection. So on the left there, this is a classic immunohistochemical stain there. The stain, when you build up the antibodies, the stain is a brown stain, and you see lots of brown expressing cells, and they're cells that are showing to be expressing toll-like receptor. So within that periapical granuloma, those cells are expressing that there. So these are obviously from biopsied specimens with apical surgery. Interesting, we always find the periapical cyst or radicular cyst. Every cyst I've looked at and done this to, I've always seen cells that are expressing toll-like receptors. And what that says to me is that there's bacteria in the system somewhere that is driving the inflammatory response and also the cystic response as well. So what that indicates is that it's been driven by inflammation, by bacteria in the system somewhere. What types of cells are involved? As you imagine, lymphocytes, plasma cells, and good old nice little dendritic cell down the bottom right there, and also macrophages based on that description we saw before. And then this was Eric here, and he did a double immunofluorescent stain just to be 100% on what types of cells were involved in the periodicular tissue that were expressing toll-like receptors. So essentially what you do, you get an antibody that's will target and onto a lymphocyte, and then you put a fluorescent marker on that. So if we look top left, that's red. Those red cells are lymphocytes. You then throw in another antibody that will stick to toll-like receptors, and then put another fluorescent on it, so it's green, and then you put the two images together, and you'll see superpositioning of red and green. So you can be 100% sure of what cell is expressing toll-like receptors. For us endodontists, it doesn't matter if a lymphocyte looks like a lymphocyte, but when you try and get things published, oral pathologists, particularly in this area, really want you to be very clear on that. So I'm not going to go into immunology because it's unbelievably complicated, really, isn't it? But this is the paper here if you want to get into T cell function particularly. It's periodontal disease, but essentially we really be looking at the same scenario when it comes to pulp and periapical disease as well. So this is by a couple of staff members published last year. And essentially, yeah, very complicated really, but when we're looking at T cells, they're actually also working on keeping things healthy, as well as trying to then take care of a biofilm disease as well. So they're trying to stop biofilm forming right at the beginning, and then they have to go into other mechanisms to deal with the biofilm if it starts to form. Yeah. So it's a very interesting interaction between health and disease mediated through the particularly the T and B cells function. So you can imagine what's happening on that left-hand side in periodontal health. You can actually imagine that happening inside a pulp as well, because pulps are always being insulted by some sort of antigen leaking through the crown of the tooth. Always shown a way back by Bergenholz, etc., to show that that bacterial antigen was able to go through white spot lesions into the and interact with the pulp, cause a pulp reaction through the internal tubules, etc. It's not the bacteria; it's the antigens like LPS, for example, that does it. So these functions will be happening inside the pulp as well. Okay, so that's the that section. So that's Mount Hobson and Mount Cook, heading south on a very cool winter's day in the South Island. But 10,000 feet in a very small aircraft. Yeah. Okay, so let's have a little bit of a look at the management of the biofilm. Yeah. So what influences it? So we know that root canal infections, root canal anatomy, we know it's very complicated. But it's even more complicated once we get down into being able to use the techniques that we've got nowadays. So this study here by Poonam Verma, who's again in Sydney, practicing in Sydney, showed that this slice on the left there is the apical portion of a mesiobuccal root of NAPA 6. And you can see how it's complicated. We know it's complicated, but it's even more complicated than one would expect. And then we've got Ala Aldana, who's actually up in Darwin. She did a bit of work on that as well, showing the complexity and how we can and can't manage it. Yeah. So that's the one by Verma. There's various sections taken through the root. And when you look at that, you say intuitively, you cannot mechanically debride some of that area. Not possible. You can't get that biofilm away. So how do we manage to be able to actually get good results in the end? So of course it comes down to chemomechanical preparation, but mechanical is important as well. So for example, in that top right image there, they're so small, that delta of canals there, they're so small that no file tip will go down there. And that'll be one of those cases where you think to yourself, well, that should be 21 millimeters, that working length, and you get to 20 and you can't get any further. It's all broken up into lots of little canals like that. So how do we manage that? Just recapping on that complexity. So the classic studies to show how we gauge the complexity of a canal, we either go vein or Vitucci. Vitucci's got a bit more discriminatory than vein, but when we look at that study with Verma, which is a micro CT slices, so we're looking at six micron slices that get reconstructed in the computer to give the image. At that level, a lot of canals just can't be classified to those old classification systems. One of the other things to consider about when we're debriding the root canal system is the degree of infection of that root canal system. The coronal and mid-root area is always a bit heavier infected than the apical area. And it's the same with that radiculodentine in that area as well. So for any given tooth at any given time, in any given person, the apical dentine has got a lot less bacterial invasion into the dentine, into the tubules, than it is mid-root and coronally. Two reasons for that. One is that the infections start on the coronal, and as we saw before on the other side, it works its way down the root canal system. And so coronal mid-root, it's bigger, so you're actually going to have more biomass of cells there and able to grow in a bigger space. So you've got more cells, been there longer. But the other major thing is that the internal tubules in the apical area are always smaller. There are always a lot of them being blocked off. So normal physiological internal sclerosis. So you get less of infection in the apical area Now we really concentrate on the apical area. You get less of infection there than you do mid-root and coronal. And again, that's some of the things that we'd consider when we're looking at debriding that biofilm from that root canal system. We don't have to go overly big for an apical prep. So I always say small apical prep, but one that then will flare out into mid-root and cervical area, because you've got a much heavier infection in there. With something like a 6%, for example, 6% rotary file, give a good indication of how that would flare out to give a prep. Other thing to consider in managing the biofilm are lateral canals. And it's always a thing for postgrads in endo. You can ask them to write an essay on the relevance of lateral canals in endodontic outcome, and they'll be writing quite a bit and come up with the conclusion that they really have not shown to have much influence in the outcome at all, as long as the main root canal is prepared properly, cleaned, chemically debrided properly. So if you can do that, then the lateral canal, with the biofilm in the lateral canal, gets managed. It gets managed in two ways. So on the right-hand side. So they're relatively large, the lateral canal. So you can get a decent biofilm forming in there. But as soon as you cut off it from its main biofilm in the root canal, it loses all that interaction with the biofilm and its ability to use nutrients from that area as well. And on that image on the right, you can see as the biofilm starts to head down the lateral canal from the periodontal ligament side. Again, we've got lots of inflammatory cells coming in from that periodontal ligament. So it's the body, the immune defense system is working on the biofilm from that side, and you cut off the biofilm mass by preparing, cleaning in the pulp canal, and probably dress it. You cut off all that nutrition coming from that area and from cells, etc., and that biofilm should just then degenerate. And that's essentially why they turn out not to be a problem, because there's lots of lateral canals in teeth. But if you do that right, it's not a problem. The other area, as we mentioned, are those micrographs of the cells of the very, very small canals in the apical areas and all these little ramifications, etc., which are very small. When you look at the types of bacterial cells in them, they don't really conform to having formed a biofilm. And the main reason for that is nutritional depletion. It's very hard for nutrition to get into these small canals and it's mainly going to be coming from the other portion of the pulp and the periodontal ligament. And it's very small, and so they don't really form biofilms. It's an old word they get called like flocculations of cell aggregations, and that's what the images on the right-hand side show. And then surrounding those aggregations, again, we've got all those inflammatory cells that are coming in from the periodontal ligament. So again, managing the main canal to your ideal, to the best working length you can get, managing that takes care most of the time of all these little ramifications as well. So that's good. So these are the goals of endo treatment, as you know. So we've got to disinfect the pulp dentine complex, or the root canal system. We're then going to produce an inhospitable environment for bacteria and prevent reinfection. So the first one is chemomaniacal prep. Second one is the obturation, root filling, and third one is your coronal seal aspect. I'll just very quickly go down. So techniques-wise, we've come a long way from the step down technique all the way through to various rotary techniques, and the standardization of these techniques, which was happening around the 1960s and 70s, has led to this development and probably a better outcome. as you know, the main thing to do when you're looking at debriding the root canal is that we should be preparing the coronal portion before the apical preparation now. So something like a step back technique, where you would go down to the apex and work your way back. You don't do that anymore. We would work from the top to the bottom, coronal to apical. You debride it a lot better. This was shown way back in the 80s and 90s that mechanical instrumentation can reduce the bacterial numbers up to 1,000-fold, or 10 to the power 4-fold, but it doesn't get rid of infection all by in the canal. Infection levels in the canal like 10 to 7 or 10 to the 8 bacteria. So you can get at the most maybe a 10 to the 3 type reduction. So you're still going to leave quite a few bacteria around. And that's why we have to do chemomechanical preparation. The main reason why we're not really getting it mechanical is lots of studies have shown, and the bottom right, the image, yeah, that a lot of our instrumentation doesn't actually contact the canal wall and debride well. And that's one area that we need to look at and develop. Yeah, so rotary niti, I guess, has helped quite a lot in getting nice preps. So the best prep you want to get to debride your root canal system is a nicely tapered, centered preparation, as you know. So I'm just going to talk a little bit about instrumentation techniques. I don't types of niti instruments. I don't really go into it too much, but I'll just mention the various things where I think there's possibly quite a lot of confusion. There's so many different types of systems out there, and so I just sort of basically get myself down to 2 things in the end, which I'll come up to the end. But with the niti instruments, again, a lot of work's been done to try and increase their efficiency, and by changing the structures. That's a rough little table there of the various things that have changes have gone from conventional niti alloy right through to the newer type of max wire, and how they're described in between. I'm going to go through them a little bit and take it from there. So we all know about conventional niti endodontic instruments. The major thing, just remember, they're austenite phase. They're in the austenite phase. So it's either austenite or martensite phase. Obviously, there's another phase, but they're austenite phase. And when they're in austenite phase, essentially what we're talking about is an instrument that's super elastic. So it's elastic and springy. There. This is a study again, Robert Ayer. He's in Sydney as well. Last I heard it, and this was quite a wee while ago, 2004. But this is the very early instruments. So this was profile and K3, and just a very basic way of charting how they prepare the canal. And both of them still had a bit of inability to fully make a preparation centric. So there's a little bit, you can see here, there's a bit over removal here. This is the profile one. It's sort of transporting on the outer surface here, and not touching the inner wall apically at all. K3 was a bit better there, but there's quite a lot of transportation up in the middle area compared, compared to there. But that gives a reasonably good shape, but not a perfect shape. Then there's been various things to be done to try and make the files a bit better. So electropolishing is one. Polish the surface, you get rid of surface irregularities, and you make the instrument a bit better. So, but by doing that, the main thing electropolishing does, it gives a better resistance to cyclic fatigue. As we know, as files are working around inside the root canal system, they're getting under a lot of stress, etc. Both from a torsional point of view, but also just a fully cyclic fatigue thing. By polishing that helped a bit. So this is one of the, again by Ala, looking at one of those instruments there, the BioRace. And just looking at the data, how well it keeps a preparation on track, you can see that the numbers of mean numbers of removal of simulated dentine. Oh no, this is real dentine because this was real teeth. In the BioRace, etc., was quite small. But the more important one is when you look at the centering ratio, you can see that comparing the two types of instruments, we're not too worried about that, but you can see that they actually kept very centered. They're actually centering. So they were essentially removing material equally around the wall of a canal. Yeah, so that seemed to be getting better there. The other development is the R phase. So essentially it's still an austenite instrument, but it gets processed, thermal processed, and there's a bit of an R phase formed in it. What it mainly does is it lowers the shear modulus and it allows a twisting process. So it allowed instruments to be twisted. Up until then, NiTi instruments had to be ground. And of course, that puts a lot of strains, micro cracks on it, and leading to the potential problems of separation. So having this type of alloy, you'll be able to twist the instrument into shape, potentially take away some of that problem. And you'll find studies that will say that it's helped, and other studies that will say it possibly didn't do too much. But it all seemed to be developing in the right direction. The other one, moving on from that, was the M-wire. Again, it's an austenite, but it's where it's treated, that starts to form martensite and also an R phase. And this gives that wire, the M-wire, greater flexibility than the conventional NiTi, and good torsional properties, etc. as well. Yeah. So this study here by Vinny Massey, who's back in Fiji now. So this was looking at, particularly look at twisted file, which is on the left, and profile was the control. And you can see by these numbers here, I'll tell you numbers, the twisted file was shown to be good at keeping centered. It had a little bit of a problem in the mid region, where it was cutting on the inner wall, which is good because that's the hard wall to cut, but it looked to be a little bit excessive from that cutting compared to the other two. But when it came down to the apical portion, it's actually quite well balanced there. But these numbers here show that it is actually engaging and instrumenting that inner wall apically, which is the wall traditionally that we've always missed. Our instruments don't touch. But this combination of material and design is allowing those missed areas to get prepped. The other major difference in the alloys to me is the martensite alloy. So these are files that are in the martensite phase rather than the artensite phase. And the manufacturer are clever, and they managed to do a thermochemical treatment to reorientate all those crystals to get the material. The martensite is softer and it's more ductile than austenite. So that means it can be easily deformed, or be called pseudoplastic, or flexible. It's easily formed. It's not superelastic, so it's not working the same way as the traditional artensite files. It's not relying on the superelasticity, that springiness of a file to be able to go down canals and keep centered. So it's doing another way. It's doing it by a shape memory effect, essentially. And it's been shown to have good properties as well, the type of files. So control memory wire, it's a martensite type material, and you'll be able to get instruments made of that. Again, it's not super elastic, so it's not working the same way as an artensite one would work. And the manufacturers have also done other things. They've done electrical discharge machining, so like the high flex EVM, to make the outer surface bit tougher, so it's sharper, cuts better, but finer torque on it, etc. It's called a controlled memory wire, basically working with that shape memory type property there. So the other ones in that area working with that control memory are the gold and blue heated treated instruments. Okay, so they're martensite, so they've got the control memory effect, and there's a couple of types of instruments there I've mentioned to do there. Again, they'll have positive abilities, and they've got negative abilities as well. But one of the things I've been showing is that they can produce a well-centered preparation. So well-centered preparation means good debridement of the biofilm. So the major way that that happens is that file, it deforms inside the root canal to the shape of the canal, essentially. Okay? And then it's rotated one way or another and cleaned. So the image on the left, just to show the difference between austenite and martensite type files. So the super elastic file, it's really, you'll bend it and it'll spring back nicely, but it does have a spring back effect, or a restoring effect, is what it originally was called, a restoring effect. The bent file wants to go back to its normal shape. So when it does that, it's producing a force on particularly the outside part of the curve of that file, and that's why it tends to cut preferentially on the outside curve of a root canal. So that's a restoring force. So of course, the restoring force of an martensite file is much, much less than a stainless steel file, and that's why it's superseded stainless steel. But the martensite one, it just deforms in the canal to the shape and stays like that. Yeah, so that's the image on the bottom right. It's all bent. So I guess you've all used them, but when I first picked them up, I thought this is crazy. They're all bent and buckled, and you go in the canal and they're all swirling around and doing funny sort of shapes before they get in the canal, and then that's what they're doing. It's a basic principle of what they're doing. So if it's conforming to the shape of the canal, much less restoring force. So it's got a good ability to keep the canal centered. And the one seems to be at the moment, I don't know, it may have changed, there might be another one out, but then they've got a max wire which has got just about everything in that they've been doing to develop things. So it's got this wire, it's got both the shape memory, so it's got the martensite type property, and it's also got a super elasticity and austenite type property as well. And the XB endo shape, I don't really want to mention files, but it's about the only one that's around anyway. It's the one that does that, combines those things. And again, people have looked at it, and yeah, it does what the manufacturer says it does. It may be good in some areas and not dissimilar to other files in other areas. And what it's described as that type of file there, and I think this wiggling here is trying to describe that when it's in a canal and it's getting bent in the canal, it changes from austenite to martensite and sort of tries to conform itself to the canal shape better, so that you're debriding most of the canal. So here's a study here. I'm not sure if you can see. Talk about this is Rodriguez Amaral. He's with us at the moment from Brazil. He's here as a visiting research fellow, and he did a microbiological study with infected root canals. Looked at reciproc blue, so that's your blue martensite file. Reciproc going reciprocating motion, and then the XP endo shaper, but also the shape XP endo finisher. And they showed that the bacterial reduction was there was no significant difference between the reciprocal blue and the endo shaper. So both were as equally effective at reducing the bacterial loading in the root canal. But when the endo shaper canals were then finished by the endo finisher file, which is a smaller type, a finer file, they find a significant reduction in bacterial loading from then on from that as well. So that final finishing of that file that would go down into the canal and then conform to the shape that hadn't been touched, that seemed to get a much better result. So that was in the general endo this year. So what type of instruments there? So as you know, there's a reciprocating, which that tends to be what that's really using is the old balance force technique. And it's a great hand technique. It takes a long time to do it, but it's a great technique if you want to get down canals and minimize your transportation. So that's what the reciprocating files are doing, as opposed to the initial rotary one. And of course, they were initially designed as a crown down technique, even though balance force you could call it a crown down technique as well. But a crown down technique where you're going from top to bottom, bigger files down to smaller files there. And to my mind, what's the difference? Nothing to me. There's no difference. There's no evidence. I don't believe there's any evidence to say that either one's better than the other. Yeah, so it's really a thing of choice about that. I personally am still on the old traditional rotary. I haven't moved on to reciprocating really. Yeah, use it a bit, but I haven't found that I needed to move into reciprocating. But other guys have, but I haven't done that. So what I tend to do, breaking all that down, is that for a routine canal preparation, something that's might have a mild canal curvature, but good sized canals. So that routine. I think an austenite type file is perfectly okay to use. In fact, preferable probably to use. Any type, probably not the old classic one, but one of the modifications one. So you're using the super elastic ability to do that canal there. So they're probably the most suited to that routine canal preparation to give you a good result. But when they get more complex canal anatomy, I will go on to a gold or a blue heated martensite file. Vortex blue is my preferred one because I started with that and I haven't felt I needed to change, but it doesn't mean anything. I'll go for that one because if it's more complex canal anatomy, I've got a file that's going to start to deform, but conform to the shape of that complex anatomy, and therefore I'm thinking it's going to have less chances of transporting or getting separation or something like that. So that's where I sit with that there. So the canals do. You can get really nice preps, and the companies will show you how really nice the preps are going. But still, you still do not get 100% bacterial reduction in bacterial loading in that infected root canal system. There's still some nooks and crannies that you cannot get to. So that's why we use the chemicals. The images on the left are from Ala Aldami, and showed this nice big anastomus, or a fin type structure there. There's the main canal prep, but there's no way you would ever be able to get into that canal with any instrument at all. So I think if anything around, any instruments around, they're all good. I don't think anything is superior to anything else. But I'd say I just tend to pick two ones, one for the conventional easy canal, one for something slightly difficult. Good. So that's looking south from about 3,000 feet up, and that's Lake Wakatipu. Queenstown's down on the bottom, around down to the left a bit there. So irrigation for the chemical management. I won't take too much time on this. Ostavic said in 2014 that nothing has been shown to be clinically superior to sodium hypochlorite EDTA, so our traditional regime. Yeah. And then the Swedish Council in 2012, which had had people like Ostavic, Bergenholtz, etc., writing this scientific report. And I haven't put all the stuff in because they basically said they can't find evidence for lots of stuff in endo, but I certainly believe that here for our irrigation, there's no real way, there's nothing really been shown to be better than sodium hypochlorite. Yeah. And it kills bacteria, kills E. faecalis. That concern a while ago that it couldn't kill E. faecalis, that's really not an issue. It can do. So we can look at making the sodium hypochlorite irrigation better, having nice proper needles, nice small needles. You may activate it some way, you might replenish it some way, etc. With these types of instrumentation. So I'm sure that helps the irrigant being able to get into the nooks and crannies, and therefore start to chemically disrupt the biofilm as well. I'm going to very briefly go on to but lasers. There again, I still consider them to be experimental, but I think they might be starting to have a bit of a road heading into conventional treatment. I don't think it's here yet, but I think it might do. So lasers can kill bacteria and kill cells as well. It's all about energy absorption. But it has the potential to be able to penetrate into areas of the root canal where you and activate the irrigant particularly. But three different types of ways lasers work. There's two ablations. One, they're both cutting one, but we can use the photomechanical ablation in irrigation. Can't use the photothermal one. That's really blowing things up, vaporizing things. And then there's the photochemical effects from lasers as well. So photoactivated disinfection and photobiomodulation. So we don't use photobiomodulation to kill bacteria. It's actually used to try and stimulate healing, particularly activation of mitochondria. And we've actually got a PhD student started this year where she's going to look to see whether we can stimulate healing and ultimately healing periapical granulomas, and see whether they can stimulate healing, get it healed faster with low-level laser therapy. But the main thing we're going to use for antimicrobial action is a photoactivated disinfection or antimicrobial photodynamic therapy. There's a number of names of them. Essentially, it's two-step. Essentially, you stain the contents of a root canal, and then you shine light into it. Okay? And essentially, the wavelength of that light will react with the stain. The stain is all being picked up by, say, a bacterial cell wall. It will then blow up the cell wall, essentially. So that helps kill bacteria that way. The other type of one I mentioned was the photoablation. Now, see this wee image starts. This is one where the laser light causes the liquid to start to move in very great speeds and then form lots of shearing forces. And you see in that little laser light pulsing going on there, eventually you'll see all the bubbling fluid in there. And that's just the light causing that to happen. And that fluid, it's been shown to be moving up to 100 kilometers an hour, so over short distances, tiny distances. That causes a lot of shear forces, and those shear forces blow up, rupture cell walls. So it can be used for that as well. But the one you probably see in the advertising around is the PIPs, or whatever, where essentially it's the light is being put into the canal area, and shock waves have been formed, or waves have been formed to help move the material around and destroy the cells. So here's a paper again. This is again Rodrigo. One of his papers. See, he did it up with Stephen Cohen up in up in LA, or California, yeah. And it's a case there. Essentially, you can see that there's a lower 6 that had a couple of instruments separated in the mesial root, which couldn't get out. So prepped up as much as possible there, and then image C is the liquid, the dye being placed into the pulp chamber and root canal system, and then E, the red bit you can see up here is that the light pulsing, but you put it into that liquid, and then that laser energizes the liquid, and then gives you that antimicrobial, increased antimicrobial action there. So you can activate the, for example, sodium hypochlorite a lot better. And E and F are these 5-year follow-up in F. And yeah, I said, oh, yeah, it looks good, Rodrigo. I'm not convinced that you've got 100% healing on it yet, but it looks good. There, you can see that it's still got a little bit of periapical after 5 years. But this is the sort of things that guys are using. There's another one of Rodrigo and another staff member, Tiago Braga, just to show where people are going at the moment. This is a pulp canal obliteration case, and took CT scans, etc., and thought to see whether if you could see the canal on the CT scans, and essentially couldn't really, but had a bit of an idea that there may be one apically. So from all those scans, etc., surgical stent has been made, which is image A, which then a burr is taken down there and guided down the canal. And then hopefully what you're expecting is that you will pick up a canal when you're going down there, and then carry on doing normal root canal treatment. In this case, they couldn't find a canal, so they went all the way down, so it looks pretty heavy. But then you can see what they've done there, and see that they've blasted it with laser as well, doing the treatment to, this could be a solution looking for a problem, but this is where things are going. And then the cone beam imaging on the right hand side, and that canal was filled up with a bioceramic, so it's not GP or seal ridden. It's a bioceramic root filling there. Okay, we're heading down south further from Mount Cook now, so we're getting near the end. Root canal filling is the last part of managing the biofilm. You've got your root canal all cleaned, and that is there to fill the space so that any fluid, it's stopping fluid coming in from the periradicular tissues and giving nutrition to any areas of biofilm or bacteria that you haven't been able to eradicate. That's what root filling does. And as we know, there's lots and lots of ways to do root filling, from the traditional lateral condensation, which still is the most commonly taught procedure in the world in schools, through to your single cone, your heated GP, etc. So there's lots of ways. Essentially, I do stick with this one from what the Swedish said there. There really is not a lot of evidence whether any particular material or method for root filling gives a better treatment result than any other. I still believe, even though it's 2012, I still believe that that's the case. Okay, so the root filling, I think we've always had the little saying, it's not what you put in, it's what you take out. So it's the whole chemomechanical prep's important. So the root filling concept is its role. It's important, but you really can't find anything that would really point yourself to say this is the best technique. In my mind, the only main thing about a root filling is, this little study here that Yoga Nathan did for the masters. These are all looked at a lot of failed cases that came into the school, and tried to determine what was the commonality of them with respect to the quality of the root filling. So you're looking at density and all that carry on. But the only thing that's significant, is the working length. It's the correct working length. Voids, not important really. But the main thing is a correct working length. Correct working length indicates correct chemomechanical preparation, correct shaping, etc. So a nicely divided root canal system. So that's the main thing. So when I talk to the students, and I'll say that, you know, first thing to do is get your working length, keep your working length, and everything else will come from that. It's the most important thing. So thanks very much for listening to me, and hope to see you in New Zealand sometime. Thank you.
Thank you, Robert, for a great presentation. This is a great refresher in understanding biofilms, the tissue, as you say. We as endodontists and dentists deal with it on a day-to-day basis. It looks like that chemomechanical management hasn't really changed since I went through dent school and through postgraduate training. However, the mechanical application has changed subsequently with the new files that are coming out with the Martensite properties of files like the M wires and some others, which sort of, as you say, maintain the files center the files in the root canals, therefore removing a lot of the biofilm effectively. It's good to see that sodium hypochlorite is still the #1 chemical to be used in conjunction with EDTA, and also with other adjuncts, as you said, like lasers and activations and devices that have come up. I won't hold you up too much longer, Robert. That was a great presentation. Thanks once again. I don't think so we have any questions. So you've done such a great job that no one has got any questions.
Oh, it's pretty late in NZ, so everyone will be happy to relax, I think.
Yeah, I think so too.
That's good.
Right.
All right.
So I'll, for all the members that are there, the CPD number is 713380. So I repeat, 713380. And so now I will close the meeting. So good night to everyone. And then once again, thanks, Robert.
Okay, thanks very much. And hi to some of my colleagues there. I see Aaron and Nick are there, and there'll be a few other guys.
Yes.
Hope to see you soon.
Yes, hope to see you here and across the ditch, Robert.
Yep.
All right.
Yep.
Cheers.